So first of all, this class, it's very, it's mainly for the biophysics, but using computational method. So it's a kind of interdisciplinary course. So let's make it very informal. So if you have any questions, please just interrupt so we can discuss. Maybe sometimes even I don't know the answer, but at least we can discuss, share some ideas, and if necessary, we can do a brainstorm, do a search to find the answers. So for the first class, it's some basic knowledge of biophysics, or it's mainly biology actually. So from DNA to proteins. Because for biophysics using computational methods, so many works are done to investigate the molecular level of the proteins or biomolecules, including DNA and RNA. So in the first couple of classes, let's just focus on some background of those things. The most important thing is from DNA to proteins. In this class, we're going to talk about the protein synthesis or the gene expression. So first, why do we learn proteins? Why there are so many methods developed to do research on proteins? For example, you guys in this lab, you know how many methods we're using in this lab to study either proteins or other biomolecules. Because proteins are very important, here just show some basic protein functions. For example, okay, the proteins make up all living materials, including hair and nails. Even hair and nails, it doesn't look like protein, but still it's made of proteins. And the blood, for example, the most important thing in blood perhaps is the hemoglobin. The hemoglobin is a special protein which carries oxygen in your blood. And we know if there's something wrong with your, let me see. Okay. If there's something wrong with your hemoglobin protein, then the ability of carrying oxygen in the blood is kind of affected, then some serious diseases happened. If someone knows about this hemoglobin, there's common mutation happens to some people in their hemoglobin, and then usually the hemoglobin makes the cell to be like spherical or spherical. But if some serious mutation happened to hemoglobin, then the entire cell in the blood becomes like a very narrow or long shape. It doesn't carry oxygen efficiently. So this is common disease, which is caused just one mutation to an amino acid in the hemoglobin protein. Protein also makes brain, nerves, enzymes. For enzymes, in this lab, so far we didn't do too much research on enzymes, but enzymes are very important. So it's in the human's saliva, stomach, and small intestine. And those proteins helps you to digest the food. And also, the proteins are cellular construction workers. So huge clusters of proteins form molecular machines that do your cell's heavy work, such as copying genes during cell division and making new proteins. So for example, in our lab, one of the most important work in the lab is molecular motors, for example, kinesins. And we have introduced kinesins functions a lot. For example, the kinesins, it carries cargoes along the microtubules in the cell from one end to the other end. That's the heavy work. And another thing is the kinesin, it's used to separate the cells when a cell is dividing into two cells. So in cell division, the kinesin or molecular motor, it's very important. So for example, in our lab, the biggest found is try to research kinesin, which is involved in cancer, because as we know, the cancer is something happened when your cell start to divide itself without any control. It's the cell division is out of control, so the cell is insanely copy itself. So there will be accumulation of those cells, which becomes tumors. And if we have a method to control the cell division, or most importantly, control the molecular machine or kinesin in those cells and dysfunct the molecular machines, then those cells will be stopped copying their self. Then the tumor will not grow anymore. Then that's the plan for a big picture of using anti-kinesin drugs to control the cancer. So it's a very hot topic. There are a lot of labs are designing drugs based on the kinesin or microtubule to stop the cancer. But anyway, this is molecular machines. That's one type of protein, and also the proteins. The proteins makes up muscles, cellular messengers, antibodies. They're all proteins. So proteins are very important. And in this lab, we work a lot on protein structures, and also most important thing is protein-protein interactions, because an individual protein, it doesn't function. The function is disabled. The protein can perform its function only when it interacts with other proteins or other biomolecules. So studying protein-protein interactions, it's very important. It's one of the focus in this lab. And here shows a protein. So first of all, protein is a chain of amino acids. Let's see, in this paper, each bead here shows an amino acid. But the amino acids, when they combine into a chain of amino acids, they're losing some atoms, oxygens and hydrogens, and each two amino acids bind together, and they will lose one oxygen and two hydrogens, which is a water molecule. So once the amino acid is in a peptide or in a bond, then this amino acid is called a residue, because it loses part of its structure. It's losing some atoms. So this is a chain of amino acids, or usually we call it a chain of residues. And then the chain of amino acid can fold into a special structure, which is called protein molecule. the protein molecule maintains the structure and maintains its function. Some proteins, they have multiple functions in different structures. So proteins are composed of amino acids. There are 20 different amino acids. That's a basic amino acid. So some of you may know based on the basic amino acids, there are some other modifications which makes the amino acids to be more complicated. For example, in some of our works, we found some post-translation modifications, which means after the protein is translated into a protein. For example, shown here from the ribosome, and then there's some other modifications added to the protein. For example, one basic amino acid could be modified into a special amino acids, and then those special amino acids doesn't exist as a single amino acid in the solution. It happens only when the protein has been fully folded. Then those special amino acid has some special functions, and we will learn it later. So different proteins are made by combination of those 20 amino acids in different combinations. So usually we call this combination of 20 amino acids as the sequence. For example, this chain of amino acids or residues, it's a sequence of this protein. We call it first order structure. First order structure means the sequence. Once the sequence is determined, then usually the protein folds into a specific structure. The combination of those 20 amino acids is almost infinity. But the structures of proteins, they are not infinite. So there are a lot of sequences share the same structure, especially for those sequences which are similar. That's why there is a subject which is protein structure prediction. People use the sequence to predict the structure of proteins. Usually, if the sequence of two proteins are very similar, let's say 30% identity are found in those two sequences, usually the structure are the same. So we can say the sequences existing in nature is much more than the structures existing in nature. There are almost infinite sequences in nature, but the structures in nature, they're very limited. If you do a search on PDB Bank or some other structure database, you will find the structures are limited. The proteins are manufactured by the ribosomes. So here shows the structures of ribosome and rough endoplasmic reticulum. This is RER, rough ER, endoplasmic reticulum. Those black dots on the RER, they're called the ribosome. So it's like a protein machine. All the proteins are made from these machines. And here shows a demonstration of how these proteins are made in this ribosome or protein machine. So it involves the messenger RNA and then transform RNA, tRNA, and then the amino acids are carried by tRNAs one by one based on the message from the messenger RNA and combine them into a sequence of amino acids, When this sequence of amino acid comes out from the ribosome, it folded into a protein. We'll introduce the details in later slides. And function of proteins. So here shows some examples of the functions of proteins. It helps fighting with diseases. So if you're sick or you get diseases, make sure take proteins which helps you to fight with disease. And it also build new body tissue, and the enzymes helps to digest other chemical reactions. And the enzymes speed up the rate of reaction. And it's also a component of cell membranes. So for example, it shows a membrane here. This is a traditional cell membrane. Actually, the cell membrane is mainly made by lipids, bilayer lipids, which is shown here, the upper layer and lower layer. Can you see my mouse, guys?
Yeah.
Okay, thank you.
Yes.
So the upper layer and the lower layers, lipids, but some important things are distributed in the bilayer lipids. For example, protein molecules. Some of them are functioning as signal translation proteins. For example, if there's something happened outside the cell from the protein which functions as signal translation proteins, they can transfer the signal from outside of the cell to the inside of the cell, mainly from the configurational change of the protein in the membrane. For example, if there's something happened, if it detected some proteins outside the cell, it can bind with the signal translation protein here. And then the signal translation protein will have theory of configuration change, which starting from the outside in to the inside in. And then there was some configuration happened in the inner side of this protein. And then the signal has been transferred. So here's the structure of animal cell and then a plant cell. We'll discuss the structure of cells in details by a video. Okay, so let me share a video with you, but when I share it via Zoom, it will not be in a high quality, so I will just send the link to you, which is shown here. Chat. Okay. Okay, so this is a link in the chat. Please find this link in chat and spend 7 minutes to watch the video on your own computer. I'm not able to share it. If I share it, you will see it very blur and there's some lags. Okay, I'll stop sharing. So please finish the video and then let's continue the class. Okay, I think all of you have finished watching the video. Now let's come back to the slides. So there are a lot of biomolecules in cells. Some of them are small organic molecules. Some others are rather large, or we call it macromolecules. For example, the micromolecules include something like lipids, which is in the membrane, nucleic acid, proteins, and it's produced through the combination of multiple smaller organic molecules. For example, proteins are polymers of amino acids, and RNA or DNA can be composed of a linear sequence of hundreds of millions of nucleotides. Okay. So working with the cell, how is protein produced from the cell? There's a process, but let's talk about it later in details. Before that, let's first learn what is gene structure. So for example, this is a gene structure or DNA structure. The element of gene structure is shown here. So this is a gene or DNA. That's a sequence of DNA structure here. For example, the gene usually is very long, so it contains some frames. For example, here it's open reading frame, which can be read by mRNA, which is messenger RNA. So it can record the message from the gene or DNA, and then the mRNA will carry the message out of the nucleus and then translated into proteins. But from gene structure, not everything is translated into protein. For example, here shows some segments of gene structures. Let's see, it has some yellow part. Take this yellow part as example. In this yellow section, you will see the DNA contains some blue and red pieces, which are shown here. We call some part exon, others are intron. So this entire gene can be read by the mRNA. When the mRNA transcript the code in the gene, it will also contain the message from exon and intron. And then there's a process which is called splicing, which will delete or remove the intron information. It doesn't encode the protein structure. So this parts will be removed, and then all the blue parts or exon part will be combined together to be the new mRNA. And those information in this blue mRNA, that's the whole information for translate into a protein. So here, finally, it can be translated into a protein in the ribosome. So this is very interesting because things like not everything in the gene is useful. For example, in this case, first of all, for the blue region in the gene structure, they are not readable by the messenger RNA. So the blue part seems like is wasted. And then even in the yellow part, everything's read by the precursor mRNA, but still part of those information are seems like useless because this intron information will be cut and removed before before it's used to make proteins. So it seems like there are a lot of useless information in the gene structure or in DNAs. And if we do a comparison between species, you will see this is for E. coli. For E. coli, let's see, the dark or the black means protein coding regions. The gray means transcript non-coding regions. The white means untranscript regions, which means for white region, it means it doesn't read at all, which is the, let me see, the blue part here. The blue part is not readable by messenger RNA. And in the yellow part, it has introns, which are non-coding information. In this figure, it's shown in gray color. So for E. coli, you will see most of the information are useful, which is protein coding region. Only 11% of the information is not readable or untranscribed regions. And only 1% is transcript but non-coding regions. So this is low-level species. And if you compare it with higher level species, for example, human, uh, Homo sapien, which means human, you will see only 2% of our DNA information are seems like useful, which is protein coding regions. And most of other informations are either transcribed but non-coding regions, or it's even untranscribed regions. So at the beginning, people think it's a big waste of the genes. So we only used 2% of our DNAs to code the proteins. But later on, more and more people recognize that in those, let's say, non-coding regions, still it has some significant functions. So there are a lot of groups and researchers working on the non-coding RNA or non-coding DNAs to recognize what's the functions for those non-coding part of the genes. It might not be used in the protein coding function, but it can be used in other functions. Okay, so here let's see the structure of DNA. This is the structure of DNA. Okay, so I suppose everyone has learned at least some basic ideas of what is RNA, what is DNA, what is protein. So DNA is a polymer of nucleotides. Each nucleotide is composed of what? So here gives you the answer. It composed of nitrogen base, and then phosphate, and then sugar, which is 5-carbon sugar. Does anyone knows what's the difference between DNA and RNA?
There's an extra oxygen.
Right. Let's see. So here, this is the sugar. In the 5-carbon sugar, if you look at the DNA structure, you will find 2 oxygens here. So that's the basic structure of DNA. But for RNA, it's losing one of oxygen here. And then that's the structure of RNA. So the basic structure of DNA and RNA is just one oxygen missing in RNA. The other structures are identical. For example, this phosphate, they're identical in DNA or RNA, and this nitrogen base, they're almost identical, but in DNA and RNA, there's a replacement by one nitrogen base. We will discuss it. We'll discuss it later. So for DNA bases, we have purines and pyrimidines. So there are two types of purines, which is adenine, guanine, and there are two other pyrimidines, which is cytosine and thymine. And usually we name it by a one-letter code, which is A, C, and what else? Do you remember? So usually A is paired with what?
T.
Right. So A is paired with T and C is paired with G. So why A must pair with T and C must pair with G? Because of the hydrogen bound. So let's look at the structure of those DNA. So first, this is phosphate molecule, which is shown in a sphere here, and then sugar. Sugar is shown as what do you call it? Pentagon here. The base pair are shown between the two strands of DNAs. A and T, they can form two hydrogen bonds, and C and G, they can form three hydrogen bonds. So usually when C and G pairs with each other, the binding energy is the lowest because they form two pairs of hydrogen bond. And when A and T is paired, the binding energy is also lowest because they form the maximum bond that they can form, which is just two hydrogen bonds. If you mispaired, for example, A to G or C to T, then you can take it as some energy is wasted. So that's not the lowest energy form. Only when A and T paired and C and G paired, the energy is the lowest. However, it happens a lot in DNA or RNAs. Some pairs are mispaired. For example, C by some, let's say, accident, C might paired with T or A paired with G. Sometimes it happens. When it happens, what can we do? The nature is very complicated or elegant. When sometimes this happened, there will be some DNA repair proteins or machines which can repair the mispaired um, base pairs. For example, if it detected G is paired with T, okay, so an enzyme or a protein will come to here cut, which cut the C, cut the T and replace it by a C, then the DNA will be repaired. Actually, there's a possibility of mistakes in DNA, so there are a lot of different types of DNA repair proteins which are working on repair the DNAs. That's a big topic. If you listened to the talk of the first candidate from the physics department faculty search, which is, oh no, it's not a physics department. It's from chemistry department. The first candidate from chemistry department. So she is working on computational biology, and one of her project is DNA repair, which solves the problem of mispaired DNAs. So DNA structures. So first of all, DNA is one helix. This is a common word to remember what is DNA structure. Usually we call it what? So usually we call it double helix because they are double strand, and then it's in this form. It's in a helix structure. So it's first discovered by Watson and Crick. So here shows a picture of Watson and Crick. So that's a model, if you can see it. It's a model of a big DNA structure. They built it by some materials. Actually, before Watson and Crick, of course, they win the Nobel Prize because of the finding out the structure of DNA, which is double helix structure. But before them, there are already a lot of proposed structure for DNA. But their model is the most closed to the real structure during that time. So actually, this is Watson and Crick. One of Crick's students is actually my teacher when I was a graduate student. His name is John Yuan. So he's mixed-blooded by British and Chinese. So he's one of the students from Crick, and he works as a dean in university in UK. But during that time, there's a like collaboration between my university in China and the university in UK. So luckily, I had one course with John Yuan. So his last name is Yuan. He's from Hubei province, actually. It looks like completely a British people, but actually his last name is a Chinese last name. So he gives a very good explanation of the biology, because he was in biology department in that university. So there are a lot of stories during that time when people first find the DNA structures. It's not easy. So there are a lot of, let's say, fightings with different groups. Some groups believe the structure of DNA is this. Others think, no, it's completely wrong. It should be something like that. But during that time, we can imagine that the experimental condition at that time, it's very weak compared to nowadays. So suppose you have very limited information and you can finally combine all of those informations and
I think your microphone cut off again, Dr. Lee.